Welcome, everyone, and thank you so much for joining us today for the Huntington Society of Canada's HD Research webinar. So if you don't know, we're hosting monthly webinars that look to spotlight the incredible work being done by pharmaceutical companies who are developing therapies in HD, but also for the researchers at Canadian academic institutions whose work really helps us to better understand the biological mechanisms of Huntington disease and then to set the foundation for future therapeutic strategies. I've been delighted to see such strong participation from across the HD community, so thank you all so much for being with us today. It's my pleasure to introduce today's speaker, Thyago Cardim-Pires. Dr. Cardim-Pires is a postdoctoral researcher from Francesca Cecchetti's HD research team at Université Laval in Quebec City, and their work focuses on the molecular mechanisms that are underlying disease pathology. So Dr. Cardim-Pires describes his research as exploring the mechanisms of Huntingtin protein aggregation, with particular attention to the regions of the Huntingtin protein outside of the famous CAG tract that can generate toxic species. So by combining things like computational predictions with biochemical, cellular, and human-derived therapies, and human-derived models, his work aims to better understand the molecular drivers of Huntingtin aggregation and to identify therapeutic targets that are beyond polyQ, which is undoubtedly an area of growing interest in the field. Today's presentation will provide an overview of this research program and the key questions driving it. The talk will run for about 30 to 40 minutes if he has that, and we'll have a Q&A period at the end. So if you do have any questions for him, whether it's related to his research or to experiences in working with an HD lab, please submit those questions in the Q&A tab, and I'll read them aloud during the discussion portion. We're very grateful for Dr. Cardim-Pires for being with us today and for sharing his work with the HD community. And with that, I'll turn it over to him. Thank you.
Thank you, Alexander, for the presentation. Can you guys see the screen, the PowerPoint?
It looks good.
Okay, good. Fantastic. So thank you so much for the invitation and the introduction. As I said, I'm a postdoc here in Université Laval with Francesca Cecchetti. I started working on this project 3 to 4 years ago. I was not from the Huntington's disease field. I used to work in Parkinson's disease. And when I arrived to work in Huntington's disease, I found one unexplored part that became my project, and I'm very excited to share it with you guys today.
Thyago, we do
yes?
see the presenter view. I don't know if that's what you wanted.
Oh no, it's not.
Okay, if you clicked on this displays at the top, yeah.
Is it now?
Now I don't see anything.
Okay, just a second. I don't know what's happening. Sure. Now?
Perfect. Go ahead.
Yes. Okay. So before talking about the disease, the Huntington's disease, I would like to give just a background about the building blocks of the cell. So the cell is a complex and organized system. It needs to work perfectly in order that the homeostasis allow us to be healthy. So there are 4 building blocks that are essential: the nucleic acids that give us the genetic information and instruction to produce the shape and function of the cell and the system. The carbohydrates, they are very important for the energy and communication between cells inside and outside of the cells. As thirdly, we have the lipids, which are very important for the composition of the membranes of the cells and the signaling. And lastly, the proteins, which are my favorite group of macromolecules, because they are the effectors of the cell functions. They behave as enzymes to produce different reactions inside the cell, and also they give a structure and shape. So together, these 4 types of macromolecules, they define how cells function. And when something goes wrong in those molecules, then we have a disease condition. So Huntington's disease is a protein-based disease, and I'm going to focus on protein from now on. So proteins are my favorite type of molecules, as I mentioned, because they are a variety of species. They have different shapes, they have different size, and many, many, many different functions. So here I would like to highlight 2. The first one is the IgG antibody that I know it's familiar to everyone. because when you took a vaccine, we can stimulate our body to produce those type of macromolecules that then can identify different pathogens or pathogenic species in our body, binding to it and promoting the degradation. We also have another type of protein that is called hemoglobin that I also believe you're all familiar because it's important to transport the oxygen throughout the body from the lungs to all the tissues and get back the consumed oxygen and bring it back to the lungs so we can put it out. If you compare the size of the proteins here, they vary a lot. And here I'd like to highlight too this protein complex, which is the capsule of adenovirus. It's a huge virus that is made of small proteins here. Each color represents one type of protein, and they bind together to form this capsule where the virus DNA or RNA is inside. So protein are very important and for functioning, and to be functional, they need to conserve its shape. It's very important to maintain its shape. So now going to the huntingtin protein, this is the molecular structure of the huntingtin protein. It was generated by a computer modulation. And you can see here 4 regions that are highlighted with different colors. The first region is this green region here, which is the exon 1, is the region where you have the CAG repeat elongation, the mutation that is now in Huntington's disease. We also have this blue part that is called N-terminal, and a pink part, a salmon part, it's C-terminal, and the bridge that connects these 2 parts of the protein together so the huntingtin can have its functional shape. And the shape of huntingtin is very important and it's related to its function because one of the functions of the huntingtin is help the trafficking of mediators and organelles inside the cell. It combines different proteins that can interact to bring those signaling components like molecules, neurotransmitter, small proteins that can communicate between compartments in the cell. So if you have a problem in the huntingtin protein that connects this protein to trafficking inside and outside the cell, we are going to have a disease condition. And as I mentioned, in the Huntington's disease, the exon 1 is the primary driver of the disease. So you have the mutation in this segment here, which is the elongation of the CAG repeat. So this mutation promotes the misfolding of the protein. And when the protein misfolds, so here you can see the shape. So this is the normal size of the exon 1, so just this small part. And in the pathogenic polyQ length, when you have more CAG repeats, more glutamines are incorporated into this part, we only have a change in this small segment of the protein as well. So the mutation makes that huntingtin sticks together in a process that is called aggregation. So basically, this small part of the protein sticks together and accumulates inside the cell. So to better understand the aggregation, I brought this analogy. I think it's a very cool analysis that it's an aggregation process that we do at home. So in a normal condition, the egg, when we remove it from the shell, it's a gel, it's a liquid form, right? So when we heat it, or we boil it, we fry it, the heat makes that the albumin, which is the protein inside the eggs, unfolds, loses its normal structure. And when it loses its structure, it loses the gel-like form and it's solid. So the albumin sticks together because of the heat, and the huntingtin sticks together because of the mutation. So from a molecular point of view, we would have the soluble albumin, which has this structure that is very flexible. And when we heat it, it starts to promote a zipper-like structure that is very packed and condensed. It cannot move. So it's very hair-like structures that then promotes the solidification of the egg. So once this process is made, we cannot reverse the solid egg to bring back to be a gel, right? We cannot do that. At least we don't know yet how to do it. So what my project and many other projects related to other neurodegenerative disorders try to do is to take this solid form of the protein and bring it back to the liquid form, the soluble part, the soluble form, the functional form. So Huntington's disease then can be grouped with other neurodegenerative disorders like Alzheimer's and Parkinson's because they all have in common this solidification of a protein. So if you look inside the brain of patients with Alzheimer's, Parkinson's, or Huntington's, you're gonna find plaques which are formed by fibril bundles and those packed zip structures that I mentioned earlier that then accumulate in the forms of those fibrils. And you can see that they all look the same. If you look at the microscope, if you don't have the name, we don't know which fibril is made related to Alzheimer's, which fibril is related to Parkinson's. What differentiates the disease is that, one, the protein that causes, for example, in Alzheimer's disease is the beta peptide, the tau protein that sticks together, form those plaques, and accumulate in a region of the brain that is related to memory. In Parkinson's disease, we have the alpha-synuclein protein. You can see here the morphology of those aggregates that stick together and accumulate the substantia nigra, which is a region responsible that's very related to movements. That's where we have the classic clinical presentation. In Huntington's disease, the mutant huntingtin that is expressed throughout the whole brain that sticks together and accumulates, especially in the cortex and the striatum. So, to give you a context, then the AG mutation, the exon 1 mutation, the elongation with the CAG repeat in the small part of the protein promotes then the accumulation of these protein aggregates inside the cell. So you lose the shape of the huntingtin and that small fragment starts to accumulate inside the neurons, the neurons. And with this, we have two consequences. The first one is that the huntingtin that should be in this form, the soluble form, to interact with other proteins, do the traffic of intracellular mediators and many other functions, it's not able to do it anymore because it lost its structure. So it starts forming those hair-like structures. And those hair-like structures that I call now fibrils, amyloid fibrils, they are highly toxic to the cells. So the AG mutation makes that the protein that was once soluble, able to traffic and travel inside the cell and outside the cell, now start to form clamps that are toxic. But as I mentioned in my introduction, I'm a structural biologist. I worked in Parkinson's disease before. And when I start working with Huntington's, before arriving to Francisca's lab, I decided to take a look at the huntingtin structure to better understand the protein and see what I was going to work with. And I was very surprised that when I look at the structure, the exon 1 is a very small part of the protein. It represents only 3% of the whole protein. So 97% of the protein were until now unexplored regarding aggregation. So my question was, what if the exon 1, the small part here, is just like the tip of the iceberg? What if it's starting the process and then we have something else as the other components of the protein participating in pathology? So it's already well established and it's unquestionable that the exon 1 that has the mutation, that has the CAG elongation, it can form those clamps that accumulate inside the cell, and you have the disease phenotypes such as neuronal dysfunction, synaptic impairments, cause neurodegeneration, and then you have the clinical signs, especially motor and cognitive deficits. So my hypothesis, the question that I asked was, what if this other part of the huntingtin protein are also able to form those aggregates, those clamps, in a mixed form? And if those clamps are formed, those aggregates are formed, if they would have a role in pathology as well, if they were able to trigger disease hallmarkers. So to answer this question, I use four different approaches: in silico with computational modeling, in vitro, where I use tests if the protein is able to aggregate, and also once the aggregates are formed in a cell model. We use in vivo model to investigate the role of those new aggregates in a biological system. And we also validate our findings with human brain tissue donated from patients for research, which are very grateful for getting them. So the first part of the project in silico with computational modeling is to identify regions that may be prone to aggregation. So I'm not going to explain how those work. I'm just going to show a piece of data that we submit the structure of the huntingtin to several predictional algorithms, several softwares that could say if the protein can form those clamps or not. And we were very surprised and excited to see that all those red regions here distributed along the protein, there are also the computer told us that they may form those clamps. So that helped us try to direct the research. So we select by many other algorithms that I'm not going to give you details here, some of those small red pieces that could form fibrils to make a more deep modeling and see if they form those fibrils. So we are very surprised and also excited to understand that some of those segments are really able to form those packs. Here I'm showing the TP1, and please save this name TP1 because I'm going to show it throughout all the methodology that we use. But the TP1 by molecular modeling, the computer told us that it's able to form those zippers that I told you that are important for the protein aggregation and accumulation. So with this computational evidence, we decided to test our hypothesis in the lab if those protein segments and the full-length protein, if they were able to aggregate indeed. So to that, we use a technique that is called thioflavin T fluorescence. It's a molecule that can bind to that zipper that I told you about. It only binds if the protein is in the zipper form, in the aggregate form, and then it can emit fluorescence and we can measure and quantify. So here's just a graph showing that one of the peptides, one of the parts of the protein that we selected called TP3, were able to increase fluorescence in the presence of thioflavin T, suggesting that maybe that peptide could form protein clamps. So when you look at the microscope, at a transmission electron microscope, this sophisticated technique to visualize small structures, we're able to see that the TP3 here that gives fluorescence to the molecule can form those amyloid fibrils. You can see the hair-like structure that I showed you in the introduction. The other two peptides that we tested, the TP1 and the TP6, that did not bind the thioflavin T, they aggregate. They form those amorphous clamps. They do not form fibrils, they form those amorphous clamps. So we were wondering if those peptides that we showed that forms clamp would interact somehow with the exon 1, with the part of the protein that is already characterized to induce pathology because it is where the mutation is. So for this, we co-incubate the peptides. We put together the TP1 peptide that is away from the mutation site with the exon 1, which contains the mutation. So remember, I just told you that the TP1 formed those amorphous clamps, but in the presence of small pieces of exon 1, the Q48Cs with CAG repeats of 48, we start to see that those amorphous clamps start to form fibrils, and fibrils are known to act as a reservatory of toxic species. So this suggests that maybe the exon 1 and other parts of the protein are interacting to form those aggregates. But until now, I was just presenting data for small pieces of the protein, small peptides. And as I mentioned, the huntingtin protein is a huge protein. It has over 3,000 amino acids. The exon 1 is a small part. So here is the representation of a Q23 with the CAG repeats of 23. You can see this relative size. So we talked to a researcher, Rachel Harding. She's a structural biologist, an amazing researcher here in the University of Toronto. And she provided us with different types of huntingtin that she prepared in her lab. So she gave us to test in the lab a proteoform, a form of huntingtin that is healthy, it's normal, one with an intermediary CAG repeat, one with a pathogenic CAG repeat with 54 CAG repeat length. So you can see that the only thing that changes in the protein that we got is the size of the exon 1, the size of the polyQ, the size where the mutation happens. And we also acquire a form of huntingtin that do not have the exon 1, don't have the mutation site. So what we did with those 4 types of protein was to incubate them in the lab and use the same molecule that I presented before, the thioflavin T, that gives fluorescence when we form those aggregates. And it was very surprising for us that the huntingtin form, which is this blue one, that do not have the exon 1, once we put them in solution, it's just water with some salts, and we put it at 37 degrees for 72 hours, we could see that there is an increase in fluorescence, suggesting that those fibrils, amyloid fibrils, those pathogenic clumps are being formed, and they don't need the exon 1 to form aggregates. When we compare with the proteoform, the forms of huntingtin that has the exon 1 with different sizes, they all aggregates in the same way. We, of course, were questioning these data. Then we decided to look at what was being produced with each type of protein. And the huntingtin without the exon 1 form very characteristic fibrils. You can see here clamped, they all cluster together. While the proteins that has the exon 1, we can see some form some vesicles with aggregates on top of it, templating aggregation. The Q36 CAG repeats of 36 form large fibrils and some vesicles that start to produce small fibrils. And the Q54, which is the pathogenic form amorphous clamp and very small fibrils here that usually small fibrils, they are related to a seeding effect. They have a capacity to travel from one cell to the other, or even from one organ to the other, from the brain to the periphery, for example. So as we show that the other components of the protein, not only the exon 1, are able to aggregate to form pathogenic aggregates, we decided to test them in a human cell model. So for that, we use human-derived stem cells. So basically, we took cells from skin biopsy and we can transform them, make them produce different cell types. So in the lab, we transform skin cells from healthy subjects and AD patients that donate those skin biopsies into neurons. So you can see here, it was from a former postdoc in the lab. She established this protocol. We can produce cortical neurons, which are from a region of the brain that is affected in AD. So I use this model to see what happens with the neurons, with human neurons, when I put these new characterized aggregates inside. So for that, we use a very cool methodology. We tagged those aggregates that I produced with a green fluorescent dye. So when you put in the cell, when you put in under fluorescent light, we can see colors, we can see where those aggregates are inside the cell. So after the treatment, what we did was to measure cleaved caspase 3, that we're going to show the pictures here in yellow. If the more yellow that you see, it's a hint that the cell is dying. So here are neurons that were produced from healthy subjects. So I use the exon 1 that caused the disease as a positive control. So you can see here in healthy neurons that they have one inclusion. So this is the exon 1 inside the cell. And you can see here this yellow dot, which is the cell death marker. So it were able to trigger at small level of cell death mechanism. I also use the 2 peptides that I presented, the TP1 and the TP3. The TP1, it was very hard to find a spot that was green, so it would somehow the cell were able to deal with it. So we can see here some cell death inclusions here in yellow. But the TP3 that form the fibrils that I show you the picture, it accumulates inside the neurons. You can see here in green, the TP3, near the nucleus as well, and also outside down to the membrane. But no cell death mechanism was triggered. So somehow neurons from healthy individuals, they are able to deal with those protein clamps that can accumulate. But when you look at neurons produced from AD patients, the story is different. You can see with the treatment of the exon 1, the Q48, here in green, there is an accumulation of those aggregates. When you compare here in the ADD condition and the healthy condition, you can see that there are lots of green spots. There are the aggregates that are accumulated. And also you can see here the yellow signal that is enriched, showing that it is triggering cell death. In a similar way, the TP1 peptide also accumulates. You can see here the green spots and was able to trigger cell death. You can see in yellow cleavage caspase 3, the marker of cell death. Curiously, when you look at the TP3 that I showed here that accumulates and do not trigger cell death, I also found TP3 and HD neurons inside the nucleus. You can see here in green, but they are not able to induce a strong pathology, a strong death mechanism as the XO1 and the TP1. We did find some spots with cell death, but if you compare with the treatment with the other peptides, they are very different. So for me, this points that we need to understand better the types of aggregates they are formed by different parts of the huntingtin. And that's what we are very interested in answering. But here I was showing the data in a cell model. So we produce the neurons in the lab and we just have neurons treated with these aggregates. We decided then to go to a more complex model, which is in vivo experiments, to understand how those protein clamps would affect the brain in living models. And for that, we choose this mouse wild type, so it doesn't have the Huntington's disease. It's a healthy mouse. And we decided to inject those aggregates inside the brain in two regions, the cortex and the striatum, and see if the animal would present Huntington's disease phenotype. And the first test that we did was an open field test, which is it's a behavioral test where we place the mouse into a box and we're going to record it. So since the mouse is a very shy animal, it tends to be in the corner to feel more protected. It tends to hide from the space that he was putting in. So he avoids the center. And this we can measure the time that it is in the corners, it goes to the center, how many times it freeze in the corner, freeze in the center, and we can extrapolate if it is an anxious behavior, what type of psychiatry changes this mouse is suffering. And when we measure we performed the open field test in those mouse, we were very surprised that after 4 months, the TP1 peptide that I showed, that's the new peptide that I'm characterizing from the huntingtin, increased the freezing time, increased the time that the mouse spends on the center, suggesting that it has an increased anxiety, fear, or is under stress. The exon 1 did not elicit a significant response when compared with the untreated animals, but there is a trend. So it's starting to present at some level. It looks like it's starting to present at some level this behavioral change. But the TP1, the peptide that I'm characterizing, it's already induced anxiety behavior in those animals. So this experiment is ongoing. We still have animals that are being treated, but we're able to sacrifice a few number of animals to see what's happening inside the brains. And the first test that we did was to measure that cell death marker that I showed in yellow, but now it's here in red. So if you see more red, it's indicative that there is more lesion, more cell death happening inside the brain. And you can see here the treatment with the exon 1. You can see some red spots here showing that indeed the exon 1, it's already known it's able to trigger degeneration, cell death inside the brain. And the TP1, the peptide that I'm characterizing, was also able to induce the cell death. You can see by the increase of these red spots, which is a stainer. It's a marker of cell death. So in fact, indeed, those aggregates are inducing some response in the mouse, and these are currently under investigation. One very important property for protein aggregates to travel across the brain and even across the body to seed pathology in different parts. So we decided to check if those mouse they would have the presence of aggregates out of the injection site, away from where we put the protein. And from that, we use a fluorescent dye called AmiTracker that is able to recognize those zipper structures and be fluorescent. And out of the injection site, away from where we put the aggregates, we're able to detect those small yellow dots, which are representative, which are protein aggregates that are now far from the injection site. And for us, this suggests that one, maybe the aggregates that we injected has a prion-like effect. It can travel from the injection site to other parts of the brain, or it induces um, triggered response of aggregation in the brain. And this is also under investigation. As I mentioned, the protocol is currently it's ongoing. So we're going to have this answer in the next months. And lastly, we decided to investigate whether those new identified sequence of huntingtin pathogenic sequence would be present inside the human brain if we would find them forming clamps in the brain, in the human brain. So we decided to acquire human specimens donated for research from 4 different brain banks. I'm going to thank them by the end of the presentation. And we took small pieces of the brain, the donated tissue, to purify the aggregates. And we confirmed that the aggregates were purified. You can see that they are very diverse. They have a different size. They have like hair-like shape. They have vesicle shape. They have like a baston shape and also amorphous type of clamps. So when we got those aggregates, we took them to a special machine that is called the mass spectrometry that can zoom in into those aggregates and tell us exactly what is made of. And we're very excited again that the mass spectrometer told us that those aggregates that were purified from the human brain, they are composed of non-exon 1 huntingtin. All of those red spots here that I'm showing in this figure, they were identified inside those protein clamps, which before they were thought to be only produced by that small sequence, the small exon 1. And now you're showing that the almost 50% of the full-length huntingtin, the whole protein, is able to form those clamps, and it's found inside the human brain. One very interesting fact that in all samples that we tested to see the composition, we identified across all specimens the TP1 peptide that identified with the computational models. I show that it is able to aggregate, trigger cell death mechanism in human neurons, trigger cell death mechanism in mouse, and now we are showing that this peptide is found inside the human aggregates. So we show by now that our approach in silico identified one good candidate. We proved that these candidates, this TP1, were able to aggregate and induce cell death in neurons, also induce cell death mechanism in vivo, and it was identified ex vivo with donated tissues. So why is this important? Why do we want to continue? We need to continue doing this. Is that in the long term, we want to use a collaboration with Dr. Stefano Ricagno to see exactly how those zippers are formed. And once we have the structure of that zipper that only happens inside the human brain, we're going to work in collaboration with another researcher from the University of Cambridge, Dr. Pietro Sormani, to design antibodies that we were able to recognize only the pathogenic species and not the soluble protein. So this is our long-term goal. We are trying to get funding to continue this research, but our long-term goal is to develop antibodies against this newly identified species. So as a summary of the presentation, as it's already known that the small part of the protein exon 1 is able to form aggregates, oligomers, amyloid fibrils, and those clamps, they are able to accumulate inside the brain, cause neurodegeneration, and promote the disease. What my work is showing now is that the other part of the protein, the non-exon 1 part that doesn't have the mutation, is also able to form those protein clamps, accumulate inside the human brain, and now we are investigating neurodegeneration mechanism with cell models and disease onset with in vivo protocol as well. So this work is only possible due to our large, to all of our collaborators. I would like to give a special thanks to my supervisor, Francesca Cicchetti, and all the group. They all help somehow with this project. We have collaborators in Canada and abroad. And in Canada, I would like to highlight Dr. Rachel Harding. She is in this project since the beginning and gave us the huntingtin different forms so we could test regarding the aggregation. And I also would like to highlight and be very thankful to the brain banks, two from the US, Sepulveda, University of Washington, New Zealand from the University of Auckland, and University of British Columbia in Canada that provided us recently more specimens so we could continue this work. Thank you for your attention, and I'm going to take questions now in case someone has it. Thank you.